In eukaryotes, transcription is regulated by two types of promoters, a core promoter and a regulatory promoter. Within the core promoter, there are a number of consensus sequences that all bind different proteins, some of which bind the core transcription apparatus, and others bind other transcription factors. And so this is the core promoter, which has, regulatory sites that may differ depending on the gene. And then you have a regulatory promoter, which in this case can have multiple types of regulatory sequences and binding sites, what they're calling here boxes or elements that interact with different types of transcriptional activators. Now, coordination of these regulatory sequences can help enable coordinated gene regulation such that these two genes are regulated by a protein that interacts with this octamer element, and while these two genes are regulated by proteins that interact with these GC elements. In some cases, when you have more than one or many of the same element, this can indicate that this element has the capacity, that this protein, this activator, has the capacity to really strongly influence transcription, whereas here it's maybe turning it up, but not nearly as much as for this promoter. So putting this whole picture together, we have our core promoter and our regulatory promoter on our eukaryotic gene. We have a protein interacting with the Tata box, who is going to start recruiting a bunch of other transcription factors. This is transcription factor 2D. And along with the RNA polymerase, and together they're going to comprise the basal transcription apparatus. And I believe I earlier called it the core transcription apparatus, but it's technically the basal transcription apparatus. And then there are going to be some activator proteins that are interacting at the regulatory promoter, and that may be interacting indirectly with the basal transcription apparatus through a coactivator or a mediator. The mediator in this case is interacting with an upstream enhancer that is another regulatory site at which activators can bind. And the mediator is allowing the regulatory activator at this enhancer to interact indirectly with the basal transcription apparatus. So upstream enhancers can have the capacity, or downstream enhancers, in this case, upstream or downstream enhancers can have the capacity to interact with any gene that it's in proximity to, and these interactions are limited by insulators, which bind to insulator binding proteins and prevent enhancers from inappropriately influencing genes that they're nearby, but may cause inappropriate levels of expression if they were to interact with them. And so in this case, we have two enhancers, both of which could interact with gene A or gene B, but the insulator is promoting more interaction of enhancer 2 with gene B and enhancer 1 with gene A. Now keep in mind there was some confusion. Enhancers, I know this looks like it's double-stranded. Enhancers are only found on one strand, even though these proteins sit down on both strands. Okay, so only one strand is recruiting proteins to that site. And again, also genes are only encoded on a single strand. And so gene A is on the minus strand, and it's being transcribed to the left. Gene B is on the plus strand, it's being transcribed to the right. And so keep that in mind. Enhancers can be on either strand, it doesn't really matter. It's also important to note that just as there are enhancer regions, there can also be silencer regions that bind silencing proteins that turn down transcription rather than turning up. And these regulatory regions can include binding sites for both enhancer proteins or silencing proteins.